Good afternoon, everyone. Thank you for joining us for the 31st ASCLD FRSC Lightning Talk episode. I am Tracy Dawson-Green, and I will be your moderator. Today's session will feature 3 speakers talking about the use of 3D technology for firearms analysis. The Lightning Talk series was developed to provide a platform for brief presentations that allow the community to share opinions, spark ideas and collaborations, highlight recent research, and convey information. The format of the session today will feature 3 speakers, each with approximately 7 minutes. Following the presentations, we will have a brief question and answer session. If you have any questions during any of the talks today, please use the Q&A feature to submit your question, and I will read them to our speakers at the end of their talks. So as I mentioned, today's episode will feature 3 scientists to talk about 3D technology for firearms analysis. That includes Johannes Soons from NIST, Xiaoyu Zheng from NIST, and Thomas Renegar from NIST. So I will turn it over to Johannes Soons from NIST, who will begin with his talk on 3D surface metrology for forensic firearm and toolmark analysis.
Thank you, Professor Green. I'm having, of course, a bit of trouble advancing the slides.
Did you move the mouse? It says it's waiting for you to control. Okay, there you should be able to control.
Okay. Sorry about that. Good afternoon. My name is Johannes Soons, and in this presentation, I would like to give you a brief overview of the advantages and some of the challenges on the application of 3D measurements of tool marks for firearm and toolmark analysis. Before I get started, the usual disclaimers. Any brand names are for illustration only and do not apply and imply endorsement, and the opinions in this presentation are mine and not necessarily the official position of NIST. So the workhorse of firearm and toolmark identification is, of course, the 2D light comparison microscope. And the images that are acquired by such a microscope are basically an indirect impression of the underlying topography of the sample. And the images are very much affected by the lighting conditions and other experimental factors like focus. And examiners use that to their benefit because they can use lighting variations in lighting conditions to highlight certain features of interest. The challenge is, though, that once you acquire the image, you cannot adjust lighting anymore. Also, there is a high degree of variability in the images acquired, which can make numerical comparisons more difficult. 3D microscopes, on the other hand, allow you to directly measure what is of interest, which is the topography, the peaks and valleys of the sample. The image data that you acquire is much more reproducible, and that allows you to basically measure once and then compare as often as you want, for example, using virtual comparison microscopy. The high repeatability of the data also facilitates numerical comparisons, as Alan will show. But there are some challenges in the application of 3D microscopy. Forensic samples are very difficult to measure. They have large slopes, deep impressions, and reflections. And furthermore, we need to put a system in place to ensure the quality of the data that is obtained. So where are we in the application of 3D toolmark images? It has been common for close to a decade in database search, like in the NIBIN system, although it's still augmented with 2D images. Virtual comparison microscopy has been introduced in court, and it has passed at least one Daubert hearing. use of 3D image data for objective identification, algorithmic-based identification, and strength of evidence estimation is still a few years out, but we're getting closer, as Alan will explain in the next presentation. So what is virtual comparison microscopy? Well, it's basically the same as light comparison microscopy, except that the comparisons are performed on a computer screen using image data that was previously acquired. And the options that the examiner has are very similar to traditional light comparison microscopy. The images can be moved, they can be rotated, lighting conditions can be changed, but the underlying data has already been acquired before the comparison is being done. And of course, the comparison is still subjective in nature. Now, the advantages of virtual comparison microscopy are, first of all, you only have to measure once, which could be done by a technician. And then you can compare as often as you like without needing access to the actual physical sample. The visualization is much more reproducible. So if you want to recreate your lighting conditions later on, that's easy. And VCM makes it much easier to do training, proficiency tests, and validation studies. However, there are challenges. And the first one is how to select the appropriate instruments. There are quite a few instruments out there that can do measurement of 3D topography. Some of those are focused on forensic applications, but some are generic surface texture measurement instruments. And in the latter case, you have to make sure that the instrument has the proper sample holders as well as access to forensic software like VCM. Instruments that are designed for forensic applications can differ in their application range. Some can only measure cartridge cases, some can only measure bullets, some can measure both. If an instrument can measure a cartridge case, it may only be able to do the head stamp and primer region, whereas other instruments can also measure chamber marks on the side walls of the cartridge case, etc. So there are differences in the capability of the instruments. Instruments can have different measurement principles for 3D measurement. Lateral resolution is, of course, important. Access to forensic software and the capabilities of that forensic software is important. Level of automation is important. Some instruments can measure an array of multiple samples. For bullets, some instruments provide automation so that you can measure the whole circumference of the bullet in one setup. And of course, overall measurement time is important. Last but not least, the accuracy of the instrument and the associated environmental requirements of the lab. In the insert in the middle, you see the same striated mark that was measured by different instruments. And you can see that some instruments have a higher noise level and that there are other differences between the instruments. And comparison studies like these are being conducted. And I can say that instruments are slowly moving towards more common characteristics for the data acquired. Now, when we talk about accuracy, the way that ISO sees it is, and these are standards for generic 3D Toolmark measurement, the way that ISO sees it is they define metrological characteristics, which are basically characteristics of the measurement instrument which can influence the result of the measurement. In other words, which can cause errors. And those characteristics may require calibration or verification in the lab. They may be used in uncertainty analysis, and the associated performance parameters can be used for instrument selection. However, we have several challenges in this field, one of which is that the measurand is not that well defined. We don't have a nice collection of minimum instrument performance requirements that have to be met. And generic artifacts for 3D topography measurement instruments may not be applicable to forensic instruments because they don't fit the sample holders or the workspace. On the other hand, the big advantage in forensics is that comparisons can be forgiving for many of the error sources. So all those metrological characteristics that are listed in the orange block, a lot of them may not be relevant when you do comparisons. For example, the vertical scale of your measurement is extremely important in industrial measurements of roughness. But when you make comparison between samples, examiners tend to automatically adjust for the differences in the overall vertical scale of the feature seen. Lateral scale may be very important though. So it depends a little bit on the type of marks that are being measured. So what is the proposed implementation approach for VCM in forensic labs? The first step is, of course, the overall generic VCM developmental validation, where we evaluate whether VCM meets the criteria necessary for application in forensics. And there have been several studies done, and they indicate that overall VCM is as good or better than light comparison microscopy for the studied scenarios, and sometimes results in fewer inconclusives. The second step is to select an instrument and to make sure that it's calibrated or that the calibration constants have been verified. And for that, NIST and other organizations are developing special artifacts that make this easier. The third step is the development of quality control charts that track changes in the similarity of measurement data obtained for an artifact that is sensitive to many error sources. And an example of those are the NIST standard bullet and standard cartridge case. So you can use that to see if significant changes are occurring in your procedures or in your instrument. And the most controversial step, step number 4, is deployment validation. And there are differences of opinion how that should be done at the lab level. One proposal currently being worked on is to do this as a mini black box study. But the challenge is that the statistics are working against you, because if you want to prove, for example, that your false positive error rate is between 0 and 2%, with 95% confidence, you need to run 150 known non-matching comparisons, which can be quite a lot for a small lab. So we're studying the ideas of pooling labs that have similar instruments and SOPs, and maybe other methods for deployment validation. So my last slide is a list of standards and guides that may help. At the lab level, the most important ones are the NC-ASB standards, as well as the best practice guides being developed by the TWIC. And with that, I will hand it over to the next speaker.
All right, great. Thank you. Thanks, Hans. Our next speaker is Alan Zheng from NIST, also from NIST. He will be talking about a reference population database for firearm Toolmarks. Alan?
I got to unmute myself, and I don't have that option if I remote controlling. All right, so hi, my name is Alan Zheng. I'm a mechanical engineer at NIST. And today, Hans went over some of the initial work when it comes to measurements and metrology and how we're advancing the science forward. And we've gotten to a point now where there is a need to develop reference population databases of firearm toolmarks, similar to what DNA did in its infancy. And that's why the acronym RapidFit is appropriate here. Same kind of disclaimer, but the motivation is simply to address these reports that have come out and will come out in the future about the subjective decision process for pattern evidence and the lack of any kind of estimation of error rates when it comes to a conclusion that examiners testified to in court. So this is what we're trying to address by using more automated methods and statistical calculations to be able to support the examiner's conclusion in court. So this project is a collaboration between the FBI, NIST, and the Netherlands Forensic Institute, or NFI. We got together about 6 years ago. FBI obviously have a lot of expertise in firearms and a vast reference collection of firearms and ammunition to help us build this database. We've developed many different types of algorithms within NIST, and NFI also has their own algorithms as well as a programming support base. So we got together to try to build this software to enable RapidFit. And what is RapidFit? I want you to imagine it as one giant Excel lookup table where we're really just trying to sample when two ground truth samples come from the same firearm, what kind of score do we get? And when they come from different firearms, what kind of scores do we get? So you can see in this first row here, we have two firearms. This is the known matching comparison. We also keep track of all of their class characteristics, things like caliber, make, model, and those kind of things. And of course, next to that is the cartridge cases that they generate. We keep track of their class characteristics as well, because we know for a fact that given the type of ammunition, you may have very different types of markings and repeatability of markings, so that can affect your score. And of course, at the end, you have some sort of score. Now, you would do this thousands of times for known matching samples and known non-matching samples. You can start to statistically model, given different relevant populations, what type of probability do you observe a particular score. So the important part of that relevant population is, of course, we need to be able to track what exactly did this measurement come from? What kind of firearm did it come from? What kind of ammunition? And the different types of class characteristics that a firearm and toolmark examiner typically document in their reports. And this helps us to be able to essentially filter through a very large database and pull out the relevant samples and scores that's very similar to what the evidence is. So the algorithm we're using as part of the similarity metric here is something called CMC. And in the very quick description, the more CMCs you have, the better its similarity. So in this particular case, you have an example of a known matching comparison. And the next example is an example of a known non-matching comparison. So again, more CMCs, the better similarity you have. So once we've developed this algorithm, you can apply this to many different firearms, ammunition types. And in a perfect world, you would hope that all of your known matching comparisons have a high comparison score on the x-axis there. And all of your known non-matching comparisons would score very low. And the y-axis in this case is just the probability of observing a particular score. So of course, that's the perfect world. And in the real world, it's a little bit messier. Sometimes your known non-matching samples can score higher, and sometimes known matching samples can score lower. And if you establish some sort of threshold in your comparison score, in this case, the green dotted line, anything to the right is an ID, anything to the left is an exclusion. You can then calculate things like false positive and false negative error rates. Now, this gives you a good overall sense of the performance of your system. However, it doesn't really tell you very much about your particular piece of evidence that you're analyzing. So right now, the field is moving towards something called score-based likelihood ratios, where using those same distributions, the blue and red curves you just saw in the previous slide, instead of establishing a threshold, we're looking at a particular score. So this score is generated by your evidence that you recovered at the crime scene compared to a test fire from a firearm, for example. You generate a score of S in this example. And all you're looking at is, given the background population has similar types of class characteristics as your evidence exhibits, what is the probability of observing that particular score given that it came from known matching comparisons, divided by the probability of observing that score S given that it came from a non-matching comparisons. And this ratio is what we call the score-based likelihood ratio. And it goes from a scale from 0 to infinity, and the higher it is, the more support you have that particular cartridge case or bullet came from the suspect's firearm. So we've tested this protocol on 4 different populations of firearms. And as you can see, visually, you do notice some differences, especially in the known matching comparisons from these 4 different manufacturers of firearms. And this is to be expected. Some firearms are a little bit more repeatable, and they have more gross marks on the breech faces, and you can actually transfer a lot of those marks onto the cartridge cases. Some other firearms are a little bit smoother, so you end up not being able to transfer as many of those individual characteristics. But one of the important things to note about this particular research is how low the known non-matching comparisons are. That means anytime there's a ground truth non-match, it's a very low probability, or it's a very tight distribution at the very end there. Now, going back to the relevant population discussions, do we really need to dive deep into very specific populations? So using those same 4 populations we just observed, we combined them, all 4 of them, into an all population you see in the red there, and we separated them out based on color and their different manufacturing. You can see that with the exception of Glocks, the other 3 populations of firearms are very similar in their likelihood ratios. So in those cases, we may not need to be very specific about which population we sample because they all yield very similar results. So this is an ongoing research topic moving forward in the future. So currently the effort is into the data acquisition. You can see a picture of the FBI's reference collection and their measurement efforts. Each one of their firearms are being test fired using this list of ammunition to really cover all the variety you would see. And in the future, the way we foresee the framework working is if a requesting forensic laboratory will submit their measurements along with the class characteristics to FBI. FBI will crunch the numbers to rapid fit and then send back a weight of evidence report to a requesting forensic laboratory. This is very similar to the CODIS database that FBI already runs. So with that, I am done. Feel free to email me if you have any questions. Thank you.
Thank you so much, Alan. We appreciate that. Next up, we have Brian Renegar. Brian's going to talk to us about objective comparisons of printer marks on 3D printed firearms components.
Okay. See if I have control here.
You do.
Okay, how do I advance the slides? I don't see them moving.
You should be able to use the arrow button. There you go.
Oh, there we go. Okay. Okay, good afternoon. My name is Brian Renegar, and today I'm going to talk about objective comparisons of printer marks on 3D printed firearm components. So this is relatively new research that we've been working on and are going to be continuing to work on for several years, probably, at least. We're in collaboration with the FBI lab, so they're helping quite a bit in some of this research. And basically, we're trying to answer the question of can a 3D printed part be linked back to the 3D printer that it was produced from? So you can see a 3D printer on the left-hand side here. Let me get my laser pointer. So this is a typical 3D printer that one might see these days. It's based on an FDM technique, so it's fused deposition modeling. And basically the way this works is that there is a heated nozzle that melts and extrudes a plastic filament and deposits that material onto a print bed surface or a build plate, one layer at a time until the part is finished or it's been produced. So there are many chances for Toolmarks to transfer from the printer to the printed part, and those are the kinds of things that we are looking at. So we can advance the slides. Kind of slow. Okay, sorry. So here's the same disclaimer that you've seen before, so I won't go into detail with that. Basically, why is this research important? Well, investigators are starting to see that 3D printed firearms or components are now being recovered as part of criminal investigations. And so, like I said, if a printer can be linked to the 3D printed part, then that may be important information as part of a criminal investigation. So 3D printers are also being used for illegal modifications of firearms, such as Glock switches or auto sears, and they're also being used in ghost gun factories these days, so illegal distribution of 3D printed firearms. So when we look at the surface topography or the surface features of 3D printed parts, there are some definite parallels to firearm analysis where we may see class and individual characteristics. So the class characteristics that you may see, these are features that may indicate the type of 3D printer that was used in printing a part. So anything from like the print lines that you can see here from the nozzle tool path. Sometimes the materials being used can tell you something, or the layer heights, the width of the print lines. These are all things that could potentially tell you something about the type of printer that was used. But just with firearm analysis, these are class characteristics, and they can only be used to exclude certain classes or types of printers. On the other hand, individual characteristics are features that may be specific to a particular printer. So that may tell you something about the specific printer that was used to produce a printed part. And these can come from a variety of sources, one of which is the build plate or the print bed surface. The random surface topography of that plate could be transferred to the bottom surface of the printed part. You can also get individual characteristics from nozzle marks as well. And these could be impressed or striated as the nozzle is moving across those top or upward facing surfaces. And there's a number of other areas as well that you may end up with individual characteristics. So we designed an experiment to look at some of this and see if it's possible to identify where a part was printed on a build plate. That's where we started out. So looking at the random surface roughness on the build plate, is it random in nature? And can you tell where a part was printed on that build plate? So we designed a small part here that has a flat bottom surface that contacts the build plate. And we printed these in 4 different locations on the build plate. And then we printed several samples consecutively at each location. So we ended up with a number of samples. Once they were all printed, then we used a 3D scanning optical microscope to scan that bottom surface of the printed parts. And from that, we were able to obtain the 3D surface topography of those surfaces and to investigate the structure here to see if it is random in nature and if it repeats from one print to the next print. So we performed some correlation analysis on these surfaces, and in this case, we're using the aerial cross correlation function. And in the top set of images here, we have 2 surfaces that were printed at the exact same location on the build plate. And what we can see is when we run it through the correlation analysis, we get a high correlation score of about 88%. A perfect score would, of course, be 100%. So it's very high then. And if we look at 2 surfaces were printed at different locations on the build plate, we see that when we run that through the correlation analysis, we get a very low score. In this case, we get a 4% correlation. So when we combine all the data together, a similar graph to what Ellen was showing, we can look at the distributions of the known matches and the known non-matches. And we can see that there's nice separation between the 2 populations, which means that the technique is working, that we're able to identify where that printed part was printed on the build plate relative to the other samples. And so the hypothesis is that this extends to other build plates or to other printers. So meaning if the surface topography is random in nature from one location to another, and you can identify the location on the build plate, then the same should be true for identifications with a particular build plate it was printed on. So one printer versus another printer. But of course, we need to do more research on this in the future here to confirm this. So we also looked at some nozzle marks. And in this case, we can see some nozzle impressions that were made. And we have 2 parts here where the nozzle lifted off of the surface as it was done printing the parts. And we can compare the nozzle impression that was left on this part versus this part. You can see that it was not a complete nozzle impression. We only got half of it here, but there's still some useful information in here that looks like it's correlated nicely between the 2 parts. So in this case, we used the CMC analysis, or congruent matching cells analysis, which is, as Alan showed you, breaks the surfaces up into small cells, and then the cells are correlated against each other. In this particular case, we got a CMC score of 14. which is pretty good considering the small region area that we were able to use for analysis. So I know I'm out of time. So one last slide here just to show you the kinds of similarities between samples that you can see. This is a striated mark from a nozzle on two different parts here. And you can see the good similarity between these two in this VCM analysis image here. So you can see a lot of correlation, a lot of good marks that look like they're repeating very well. And that's it. Thank you. I will relinquish control there. There we go.
Thank you so much to our speakers today. We're running short on time, but if you have any questions, please put them in the question and answer area, and I'll read them to our speakers. We do have a couple of questions. First, I'm not sure, I think any of you, this is directed to anybody that wants to answer. Do you think that the 3D printed firearms analysis needs will increase in the future? Do you think you'll see more of them in future casework?
Yeah, I can take that one. Yeah, we think that 3D printing is going to become more popular for criminal activity. The technology has improved quite a bit over the years, and it's definitely more affordable than it used to be. So we think that a lot more people are going to be using it in the future. Not to mention there's been quite a few changes to laws and regulations, both federal and local, in terms of 80% kits and things like that, possibly making them a little bit more difficult to purchase. So we think that it is going to become more attractive for 3D printing for criminals to use in the future. So that's one of the reasons why we think it's important to continue the research that we're doing.
Great, thanks. Also, what do you guys feel like the role of AI will be in this field going forward?
I'll take that one. I think obviously AI is only going to get better as time progresses, but it has very severe competition with the capabilities of examiners. I think the key to AI will be transparency. For example, in the databases that are being used, as Alan described, but also transparency in how it derives its result. So basically, explainable AI will be very important for applications such as firearm and toolmark identification. I think near term, I see applications of AI like in image segmentation. For a firearm, for example, you need to extract the firing pin impression and separate it from the breech phase impression or the aperture shear. I see it in applications such as recognition of class characteristics. Long term, I don't think it will replace an examiner, but it will may be able to provide a very solid second opinion.
Great, thank you. This next question is for Alan. Will the likelihood ratios be relegated to a single machine or be ubiquitously applicable across all VCM platforms?
Thanks. Yeah, it will be applicable to 3D systems out there and in the future as well. Once we get the framework set up, a laboratory with a 3D system, they have to demonstrate a couple of quality assurance measures that we'll spec out. And as long as you're able to meet those specs, then you should be able to submit the measurements along with the class characteristics to FBI, and they will be able to calculate those likelihood ratios for you. And the reason we want to centralize this effort is because to build a reference population takes a lot of effort and resources. And also, if you have a bunch of, let's just call it shoddy reference populations out there, you can have very different results that are not supportable. So that's why we're trying to build a very large and diverse population that everyone uses to calculate their score-based likelihood ratios. And of course, any future updates will continue to build and expand the reference populations as well as we move forward.
Great, thanks, Alan. That's all the questions that we have for you guys today.